Some peptides are claimed to dramatically increase muscle growth, boost recovery, and raise growth hormone levels. But what do these peptides actually do inside the body? And which ones are actually connected to muscle growth pathways? GHRP6 is a synthetic hexapeptide designed to trigger the body's own growth hormone release. It binds to the ghrelin receptor called GHSR1A on the pituitary gland. This action triggers a rapid surge of intracellular calcium, starting a chain reaction that produces a strong, pulse-like spike in growth hormone levels. The downstream effect is an increase in circulating IGF-1, which plays a direct role in muscle protein synthesis and recovery. In small human studies, a single dose of GHRP6 can raise growth hormone levels up to 4 times baseline, and IGF-1 can remain elevated for days. What sets GHRP6 apart and makes it a staple on bodybuilding forums is its pronounced appetite-stimulating effect. It often leads to intense hunger soon after use. This, combined with its reputation as a cost-effective, research-only compound, has fueled its popularity among those looking to maximize bulking phases. However, it is important to note that while GHRP6 is widely discussed for its muscle-building potential, its legal status remains firmly in the research category, and its long-term effects are still not fully understood. Ipamorelin is a synthetic pentapeptide designed to trigger growth hormone release with remarkable selectivity. Unlike older secretagogues, it binds to the same ghrelin receptor, GHSR1A, but does so in a way that minimizes unwanted hormone surges, particularly prolactin and cortisol. In practical terms, this means ipamorelin produces a clean pulse of growth hormone without the side effects often seen with other compounds. The result is a moderate, steady increase in circulating IGF-1, supporting muscle protein synthesis and recovery while avoiding water retention or increased appetite. Because of its short half-life and targeted action, ipamorelin is often described as the most refined option among growth hormone secretagogues. Its reputation for minimal off-target effects and smoother hormone profiles has made it popular for those seeking lean mass gains without the complications tied to broader-acting peptides. Despite being banned by the World Anti-Doping Agency and available only as a research chemical, ipamorelin remains a frequent topic in discussions about safe and effective peptide protocols. Hexarelin is a synthetic hexapeptide designed to deliver the most powerful growth hormone spike among the growth hormone-releasing peptide family. It binds tightly to the ghrelin receptor, GHSR1A, on the pituitary gland, unleashing a rapid and pronounced burst of growth hormone. This surge quickly elevates circulating IGF-1, activating the PI3K ACT-mTOR pathway in muscle cells and supporting protein synthesis and repair. However, hexarelin's potency comes with a catch. Repeated use leads to fast receptor downregulation, meaning the body adapts and the growth hormone response fades within weeks. Studies show growth hormone output can drop by 60% after 3 months of continuous dosing, but it rebounds after a break. There is also a unique cardiac angle. Hexarelin activates ERK signaling in heart tissue, which may be beneficial at low doses, but it raises concerns about long-term cardiac remodeling if abused. For bodybuilders, hexarelin's appeal lies in its unmatched growth hormone pulse, but its rapid tolerance and potential heart effects make careful cycling and caution essential. CJC-1295 is a synthetic peptide modeled after growth hormone-releasing hormone, but with a key twist. Its structure resists breakdown in the body, allowing longer activity. The most significant innovation is the DAC version, which stands for Drug Affinity Complex. By attaching a special chemical group, scientists enabled CJC-1295 to bind albumin in the bloodstream, stretching its half-life from 30 minutes to as long as 7 days. This albumin binding trick means a single weekly injection can keep growth hormone levels elevated, mimicking the body's natural pulsatile release through the CAM-PPKA pathway. In practical terms, the DAC version allows for less frequent dosing compared to the original, which requires daily administration. This engineered pharmacokinetic profile has made CJC-1295 a mainstay in performance circles where convenience and sustained hormone elevation are prized. Its reputation as a set-and-forget peptide continues to drive interest among those seeking steady support for muscle repair, fat loss, and recovery. Tesamorelin stands out as the only peptide in this group with full Food and Drug Administration approval, but its legal use is limited to treating excess abdominal fat in HIV-associated lipodystrophy. Structurally, tesamorelin is a synthetic growth hormone-releasing hormone analog, modified with a fatty acid chain to extend its half-life to about 12 hours, allowing for convenient once-daily dosing. It binds to the growth hormone-releasing hormone receptor on the pituitary, triggering the cyclic adenosine monophosphate pathway and leading to a steady elevation of growth hormone and a moderate rise in insulin-like growth factor 1. In large phase 3 clinical trials involving people with HIV, daily tesamorelin injections reduced visceral abdominal fat by about 20% over 6 months, with a small but measurable increase in lean body mass. These results are often cited in bodybuilding circles, but the context is important. Tesamorelin's approval is for a specific medical condition, not for general muscle growth. Off-label use is illegal, and it is banned by the World Anti-Doping Agency in all sports. Its reputation for clinically proven fat loss and modest anabolic support is grounded in high-quality evidence, but its role in healthy athletes remains untested. IGF-1-LR3 is a laboratory-engineered version of insulin-like growth factor 1, designed with a 13 amino acid extension that allows it to avoid most IGF-binding proteins in the bloodstream. This modification stretches its half-life to about 20 hours, making it far more stable than the native hormone. Unlike peptides that work by stimulating growth hormone, IGF-1-LR3 acts directly at the muscle cell by binding to the IGF-1 receptor. This triggers the PI3K-AKT-mTOR pathway, a central driver of muscle protein synthesis and satellite cell proliferation. The result is a strong anabolic signal that can promote muscle fiber growth and help prevent muscle breakdown. Because it bypasses the growth hormone axis entirely, IGF-1-LR3 is often discussed as a way to deliver direct muscle-building effects. However, its insulin-like activity can lower blood sugar, creating a risk of hypoglycemia if not managed carefully. Its reputation for potent, receptor-driven muscle gains has made it a topic of ongoing interest in performance circles, especially among those seeking results beyond what growth hormone secretagogues can offer. PEGMGF, or pegylated mechano growth factor, is a modified splice variant of IGF-1 that stands out for its role in muscle repair. Unlike systemic IGF-1 analogs, PEGMGF is released locally in muscle tissue after intense mechanical stress, such as heavy lifting. Its structure is altered by attaching a polyethylene glycol chain, which extends its half-life to about 6 hours, long enough to remain active during the critical post-workout window. PEGMGF binds to the IGF-1 receptor on satellite cells, triggering the PI3K-AKT pathway and driving satellite cell proliferation. This process expands the pool of cells available for muscle repair and future growth. In animal studies, repeated PEGMGF dosing led to increased myoblast numbers and modest gains in muscle fiber size. Its appeal in bodybuilding comes from the idea of accelerating recovery and supporting adaptation by targeting local muscle repair rather than just boosting systemic IGF-1. Weekly dosing trends are common among those experimenting with research-grade compounds, but clinical data in humans remain limited and legal use is restricted to laboratory research. Follistatin 344 is a 344 amino acid protein known for its ability to bind and neutralize myostatin, the body's main brake on muscle growth. By blocking myostatin, follistatin 344 lifts the limits on muscle cell proliferation and protein synthesis, releasing the SMAD pathway's hold and allowing the ACT-mTOR axis to drive unchecked hypertrophy. In animal studies, this has led to dramatic muscle mass increases, up to double in some mouse models. But in humans, the picture is far less clear. Early gene therapy trials showed only modest gains and raised serious red flags, including disrupted reproductive hormones, cardiac remodeling, and potential tumor risks. Despite its reputation in bodybuilding circles as the ultimate myostatin inhibitor, follistatin 344 remains strictly experimental, with safety and effectiveness in healthy adults still unproven. Peptide science keeps evolving, but unregulated use still outpaces clinical research. The line between progress and risk is razor thin, and for now, every choice remains an experiment. Let me know your thoughts below.